Well, I would like to welcome our today's speaker, Professor Enrico Dalla, from University of Sheffield. And he for a long time was working towards understanding the properties of these musculoskeletal tissues. And it was different type of research which were based on modeling and simulation, as well as also on analysis of data, especially using these pattern recognition methods. And this is the most important what Enrico is doing. And he is a professor of multiskeletal biomechanics at the University of Sheffield. And also he is the director of a skeletal AI laboratories. So he is member of many European bodies, and also he's chairing one of this body. And his research was well recognized by obtaining many European grants. And in 2024, he has been selected as ERC, which is very difficult to obtain, consolidator. And I think that that's maybe in short what I can, how I can introduce Enrico. During the talk, if you have a question during the talk, please ask this question on, you can use for these questions or chat, and I will transfer your question to Enrico. Besides of this, as usually, we will have questions and discussion at the end of Enrico talk. So Enrico, screen is yours.
Thank you very much, Marian. Can you see my presentation?
Yes.
So the first one, all right. First slide. So thanks all for the introduction. Yes, no, it is a pleasure to actually talk to the Sano people and the other people in Sheffield and across Europe, if there's anyone joining. And yeah, as Marian said, I mean, I work in a number of research projects, and I will talk about one of those, which is mainly on the biomechanics of the metastatic spine. And by background, I'm a mechanical engineer, and I work in the Faculty of Medicine. So my interest is every time towards both the development and the validation of the models, and also to the translation of them into clinics. Today, I'm going to talk about, in particular, the development and validation of the models. Marian said that being selected as an ERC consolidator grantee, and was very happy to do so. And indeed, it is true. And then I had to decline it because there was no UK to EU agreement. But then I got the funding from the guarantee fund from the UK to do exactly the same project. So selected by the ERC, but funded by the EPSRC, I should say. But it has been a great honor to get that project awarded. So today, as I said, I'm going to talk about computational models to improve the stratification of patients with spine metastasis. I should say that it is mainly a roadmap to this final title, because we've done a lot of work in the background during the development of better understanding of the biomechanics of metastatic vertebrae, and then looking at developing and validating computer models that we hope through a European project I will talk about towards the end of this talk we will apply them to stratify patients. So my team works mainly on bone biomechanics, and for doing that, we do both work on clinical applications, so basically the development of models to improve the diagnosis and the assessment. Actually, the diagnosis of femoral fractures or vertebral fractures, and the assessment of treatments to avoid them. We did also some work on spine fixation, and these are mainly based on computational models, finite element models based on biomedical images of the patients. And we do also work a lot on the preclinical side. So in particular, by developing tools to evaluate what is the effect of new treatments, combining animal models. So usually these are in vivo mouse studies and computational models. But I will not talk about it today. And finally, as an engineer by background, so we developed quite a number of methods, both on the imaging side, on the image processing side, and experimental tools to validate especially the outcomes of biomechanical models. And I will briefly touch, or at least show some data that we could achieve with these methods. So currently in my group, we have 4 active projects. One is the Metastra project. It is a European project that with a big consortium will try to improve the way we stratify patients with spine metastases which are at high risk of fracture, and I will talk about it later on. Then the second project is a project about interspecies digital twins to predict bone adaptation over time and improve treatments for osteoporosis with this very weird acronym. It is virtual mouse to human twins for osteoporosis. And again, I will not talk about this, but if you are interested, feel free to contact me, get in touch, and I'm happy to share what we're working on. And then we have a collaborative project with the University of Liège, Leuven, and Melbourne called the Unite project, where we look at the effect of osteoarthritis on joint degeneration. And this mainly using a combination of mouse models and the computational models and advanced experiments to characterize the interfaces. And finally, there is a sort of toy project, I would say, that was actually my project for the postdoctoral Marie Curie Fellowship when I came to Sheffield more than 10 years ago. And we developed at that time an algorithm called BoneDBC, which allows us to import 2 images of the undeformed and deformed tissue or object that we can scan with a micro-CT and provide a deformation pattern. And this is still a sort of tool that we still use through collaborations across the world. And again, if you are interested in using that, please get in touch and can tell you how we can share what we have learned so far and the algorithm. So the outline of today's presentation will be talking about a bit the motivations and the challenges associated to bone metastasis. And then I will show you some experimental work actually that we did to characterize actually the properties of metastatic vertebrae. And then 2 approaches that we have developed in the past 5 to 6 years to evaluate the effect of the lesions, on the metastatic lesions on the vertebral body mechanical properties with a sort of ex vivo application. So still in silico, but as you will see, but motivated by, so without using biomedical images from patients. And the second one is actually a tool that we are developing, a pipeline that we are developing to evaluate the vertebral strength from biomedical images. And finally, I will talk through a bit few perspectives and also tell you a bit about the Metastra project. As Marian said, please, if you have questions and you want me to stop and ask a question, write it in the chat or just jump in if you can unmute yourself, or Marian will tell me. I don't see you, so in case that you want me to stop, you need to tell me. I will not see the faces or the chat. All right, so let's start with some motivation slides. I kept it quite short because there's a lot of work in the literature that can be seen, and there's a lot of details about actually how problematic, critical is the problem of metastatic disease. So these are basically metastatic lesions made by cancer cells coming from a primary tumor, which could be in the breast, in the prostate, in the colon, in the lungs, and they all at the later stage metastasize somewhere else and create these lesions. And bone actually is a place, is an anatomical place where actually this lesion appear very often. And in particular, the spine is one of these anatomical places where they appear very often. They have a different types, so they can be lytic, so basically the sort of holes in the structure. As you can see, I don't know if you can see let me get the pointer out. So as you can see here, as sort of very low bone mineral density regions in these CT scans of the human vertebral bodies, or they can be quite diffuse as these lesions here, which are usually coming from blood cancer like multiple myeloma. And the idea is that, of course, these lesions may decrease the bone strength and increase the risk of fracture, but clinically it's very difficult to understand actually who are the patients who are at high risk of fracture. So there are some scoring systems. Actually, the more advanced is the spine instability neoplastic score proposed by AO Foundation, and basically here, what it does is that the clinicians look at different features of the metastatic spine or metastatic vertebra, I should say, and based on each of these features, they score between 0 and 3. And in the end, by this classification, it comes up that the patient or that vertebra is pretty stable if the score is very low, and therefore usually you don't do anything. Or it is very unstable for very high scores, and in this case, usually the surgeon, the orthopedic surgeon, and the oncologist then sit together to try to find what is the best way of fixing the spine. But there's a huge range which is very indeterminate, and basically the decision goes back to the clinicians and to their experience. So there's also a number of limitations of this since. So it lacks of objectivity, of specificity, and usually it leads to over-treatment, even though I have to say at least it is very reproducible. So unlikely like other scoring systems, at least it is pretty reproducible. However, as bioengineers, now the question is that can we do better? So can we create some tools that are a bit more objective, more mechanistic, and can somehow predict the risk of fracture in these patients based on the biomedical images, for example, of those patients, which usually are taken to evaluate the status of the spine, like quantitative computed tomography that you see here on the left. So in fact, actually, there are some tools like the finite element approach or FE models that I will call it FE in my talk, basically where we can convert CT images into finite element models. These are biomechanical models of that particular vertebra, of that particular subject or patient over time that has been scanned with CT. And based on the distribution of the density, we can predict the bone strength and therefore the risk of fracture. And it has been found that for osteopenic, osteoporotic, and healthy patients, or healthy subjects, I should say, this can predict pretty well, actually, the bone strength compared to experiments in the lab. And also, it can be used to classify patients at high risk of fracture, especially if this is applied, for example, to the femur. And it does also predict the bone strength better than, for example, what would do the standard clinical tool, which is a machine called dual X-ray absorptiometry or dual energy X-ray absorptiometry or DEXA, where you can evaluate the aerial bone mineral density in the lateral side or in the anterior posterior direction. And so these models are very promising. The big question is that can we apply them to study the biomechanical properties of vertebrae with metastatic lesions? The problem is that we know very little about the mechanical properties, even experimentally, of these very heterogeneous bones. And therefore, there's the need of improving our understanding of the biomechanics. There's a study done a few years ago by the group of Philip Dessay in Bern, where actually they tested some vertebral body sections. So they removed the end plates, which is this part usually that would be connected to the intervertebral discs and the other adjacent vertebrae. They removed that, and then they test them mechanically in the lab, and they could see that actually they found different vertebral strength depending on the type of lesions. So as I mentioned before, we could have a type of lesions which are lytic or lytic, similar to the ones that I showed you in the pictures, which are low density areas. or they could be blastic, which means that actually, I will show you some example in a bit, but you can see here also a typical blastic lesion, which is basically a region of hyper mineralization, so where the density is even higher than the standard healthy bone, as you can see here on the left. And this actually leads to usually a stronger vertebra compared to lytic lesions, but leads also to failure of the adjacent vertebra in many cases. Or they also can be mixed, and in this case, again, they have both features of the lytic and the blastic lesions, as you can see from the bigger range of mechanical properties of bone strength. So already this study showed that there's a lot of heterogeneity, but they missed, I think, a couple of things, which are basically the importance of testing this vertebrae with their own end plates, as I will show you in a bit, in order to better understand the failure mode. They also used this experiment to look to validate or to compare actually vertebral body, computational models of the vertebral body slices based on high resolution images. So these are micro CT images. These are not clinical CTs. These are CTs that you can get, micro CT images that you can get in the lab. And they built some homogenized models and they predicted the ultimate strength from this, and they compared with the experiments that they did in the lab, showing that if you compare them with standard or let's say from vertebrae without lesions, they could predict in a similar way the bone strength. And again, this is a bit of, there are a number of simplifications here in the fact that, first of all, this is based on high resolution images, not on clinical CTs. So they have high resolution, and I will show you something in one of the projects that we did about that. And of course, again, this is just limited on a small part of the vertebra. So there's still a lot of things to do, and there are plenty of challenges if you look at the application of these biomechanical models for the metastatic spine, starting from the creation of the models in terms of making it more general, in a way that is more generic, so including also the posterior side and the end plates, making these pipelines more automatic. Also, usually you need expertise in imaging, image processing, and biomechanics, and also doing a lot of verification, validation, uncertainty quantifications on these models. There's also a number of challenges related to the identification of the material properties, of the loading conditions, and so on and so forth. So, I mean, I don't want to list all of those, but just I put this list together, thinking about it in like probably 10 minutes, just to say how complex is still, how little we still know about this project, this problem, and what are the things that we could improve. In particular, also predicting over time how the bone will change due to the lesions and how the mechanical properties will change. And even in the end, of course, looking at effects of different interventions. So going back to the question, how can bioengineering in general, how can we help in trying to improve this scenario? I can think of two big areas here. So one area is, let's try to learn more about the bone mechanical biomechanics, the bone mechanical properties in this objects, in these very complex objects, and try to improve the scoring systems here to help the clinicians in getting a bit, maybe more quantitative and better ways of stratifying these patients, or actually using these computational models, which are very subject specific, and in that case, creating pipelines that can help and can be relatively easily be translated into the clinics. So I will start now talking about especially the first part, and then in the second part of the lecture, the talk, I will just move towards the second part. So in order to try to better understand what are the features of this vertebrae with metastasis that affect the bone strength, we need to better understand the biomechanical properties of the vertebrae with these lesions. And that is basically the goal of a number of studies that we did in my group a few years ago, together with Marco Palanca from the University of Bologna, who came to Sheffield as a Marie Curie Fellow and started his independent academic career there in the last few years. So what we did with Marco is that we said, okay, well, let's design a tool that allows us to study in detail how these bones deform under load and when they fail. So what we did is that we developed this loading device, and I can tell you here in the magnified version. You can see that this loading device fit inside our microcomputer tomography system, so we can image this object, which in this case are porcine vertebrae with induced mechanical induced lesions, and then we tested also, as you will see, human vertebrae. We can scan them before applying a load, so in their undeformed configuration. We can scan them again also after we applied some load and see how where the deformation localizes. So what is the weakest spot, basically, of the vertebra. And in order to do that, actually, we have this jig that can fit into the micro CT. It has a Perspex tube, so that actually it is radiolucent. There are two systems here to fix the sample and to embed the adjacent vertebrae so that we can apply a controlled load. We have the load is applied through a manual screw that pushes this bar here, where there is an axial bearing to remove an axial bearing to remove the rotation, and then we have an axial load cell that is used to measure the axial load. And what we wanted to do in this project is to say, look here, let's use some porcine vertebrae where we know that are pretty consistent in terms of geometry and density distributions, and let's induce mechanically some defects like this one that you can see here in the anterior side of the vertebra, and try to understand how this defect would affect the deformation and the strength of the vertebra. In order to do that, as I said, we collected images of the vertebra in its undeformed configuration and in its deformed configuration. And we use a technique called digital volume correlation to find the displacement map and then differentiate them into strain maps to see what is the localization of the strain before and after inducing mechanically this lesion, which is somehow representative of lytic lesions. Of course, this is a 3D assessment. So here, actually, it's a lateral view of this vertebra with the defect here. And you can see here, actually, these are the map of deformation around the defect that we can measure with the DVC in 3D. So we did it for different lesions in different regions of the vertebra, anterior, frontal, or lateral left or lateral right. And what we could see here, actually, it is that the induction of the lesion has changed dramatically, actually, the way the distribution of the strains is localized, basically, within the vertebra, both by looking at the frequency plot, but even more interesting by looking at the distribution of the deformation, with some unloading of the part related, in the superior and so on the top and the bottom of the lesion, and concentrating more deformation close to the lesion. We also did, and if you're interested, all the details are in this paper here in PLOS ONE in 2020. Basically, what we did is also we split the vertebra in different regions, first in different sections, so the top, the middle, and the bottom, and then each section has been split into 9 compartments to identify before and so what is the difference of strain before and after the induction of the lesion. And we could see that actually, for example, if we induce a lesion in the middle section in the anterior side, we could definitely see effect of significant changes significant increase in strains in the regions of the middle section and on the anterior top part of the vertebra, and a reduction of the strain in the blue regions. So again, here it's the first time where we looked at the effect in 3D of these defects. And we can use this information to better understand how they are affected by how the biomechanical properties are affected by the location of the lesion. But then we said, okay, well, these are porcine vertebrae, and these are probably very simplified lesion. What if we test actually human vertebrae? So we did a cadaveric study where we collected 15 spine segments. So these are from donors who died of primary tumor, different types of primary tumors, and they all had metastasis in the spine. So we could identify vertebrae with a lesion and vertebrae without a lesion that we would define as control. And we could load in our jig, similar to what we did before, but this time with 2 vertebrae, 3 intervertebral discs, and 2 half vertebrae to fix the sample in our jig. And we could identify under load what is the deformation of the control vertebrae, so which didn't have visible lesions, and the vertebrae with the lesions. And we did that for vertebrae with different types of lesions, and we could do high resolution images of this vertebrae, of both of them, during the test. And you can see here how very complicated and heterogeneous is the structure of this vertebrae, with sclerotic tissues in some areas, lytic tissues in others, and very, very heterogeneous material, densities, and therefore likely material properties. So unfortunately, we are really pretty far from this simplified lesions that we simulated before, but this gives us an idea of what happens in the real case scenario. And as I said, we have different types of lesions. So we have the controls, we have the vertebrae with lytic lesions, the vertebrae with mixed lesions, so both lytic and blastic, and the vertebrae with blastic, which are super dense here. So we did an assessment of, these are all 3D assessments here, just put example of cross sections for simplicity. But basically, we just evaluate the bone volume fraction in, so basically the density of the bone within the vertebral body, confirming that indeed the vertebrae with the lytic lesions, which are here in this oblique pattern, have lower density than controls that had lower density than the mixed ones that had lower density than the blastic ones. So this density just confirms that the classification was correct. But when we looked at the measured mechanical properties, so here we're talking about the failure load. So it is the ultimate load until the vertebra, the vertebrae, or the spine segment resisted, so before one of the vertebrae failed, or the normalized failure stress, or even the minimum principal strain, we could not see patterns by just looking at this vertebrae in groups. So we really, so the message here is that to analyze this vertebrae, you need really to look at every single component and what happens on every single case, what happens by even looking at these higher resolution images and biomechanical and advanced mechanical testing. With our approach, we could identify the areas where the lesions were, and also the strain distribution around the lesions or in the adjacent control vertebrae. And this is a case of basically a vertebrae with lytic lesions here in red that localize the deformations that you see here in orange and in green around the lesions, while in the adjacent control there was almost no localization of deformation. While in vertebrae with blastic lesions, we could see indeed, as observed in clinics, that the deformation localized in the adjacent control, while this vertebrae had basically no deformation, or these are principal strains, principal compressive strains that are plotted here from our DVC. This is confirmed also if you look at examples of vertebrae with lytic, mixed, and blastic lesions. If you just look at the frequency plots of the deformation within the vertebral body, you see that the metastatic vertebrae with lytic lesions has a much higher, or in absolute value, principal compressive strains, which means that the vertebra is more deformed. For mixed lesions, you have regions that are a bit more deformed, but a bit less than what you would expect from lytic lesions, while for blastic lesions, you have even a lower deformation in the blastic vertebra compared to the adjacent control. So again, just to say that the biomechanics behind this is very complex, and we need to look case by case at what type of lesions we have. With this approach, we could also look at failure modes. So these are basically images of the same vertebra before and after failure or fracture that has been scanned. So we're basically in red, we see the undeformed vertebra. In green, we see the vertebra deformed after failure. And in yellow, it is after the registration, is basically every part of the tissue that has not been deformed at all. And you can see here, for example, that what happens here is a localized failure of the tissue just below the end plate, which is this thin cortical bone plate that you have on top of the vertebra. In other cases where there's a big lytic lesions, for example, of course, what fails is just the end plate below the lesions, because simply there isn't a support. But we see also a failure on the other side, still in the same region where the lesion is. In some other cases as this, actually, we see how complex the deformation here is, that you can see here in green, where actually what we observe is a failure of the tissue between the end plate and the metastatic lesion, which is this big black dot that you have here in the middle of the vertebra. And in this case, of course, it leads to a big deformation of the end plate, which is extremely important. And that's going back to what we were saying before. If you do a mechanical test where you cut out the end plate, in the end, you miss probably the weakest and the most important part of the structure. So that's why it's so important to look at the local deformation of these bones. Other examples here, just to tell you that in some cases, the deformation is extremely localized. In this case, for example, just in the inner portion of the end plate, or in this part here where you have a blastic tissue on the right-hand side here, you see that what fails in the end is the portion of the tissue between the end plate and the blastic lesion, which is almost not deformed at all. So the deformation can be also extremely localized. So we can, of course, look at the failure modes, and we can quantify the deformation with our digital volume correlation approach. For example, this spine segment where there is a metastatic lesion in one of the vertebrae and the other one is a control. And you can see actually under load how big the deformations are in the middle around the lesion, and how big, and where actually this localization happened. This is just a zoom out to show that we can get a very detailed assessment of where the deformation localizes and what fails first in this vertebrae. For example, here, if you see the blue is basically where we have the highest absolute third principal strain, and that is where basically you have this failure of the end plate here, just above the lesion. Numerically, the good thing of making this mechanical testing on spine segment where we have both a control and the metastatic vertebra, what we can do is that we can look at them differentially, and we can compare basically the deformation in the control vertebra with the one of the metastatic one. And we can, here we just show that for the vertebra with the spine segments that included a vertebra with lytic lesions, in most of cases we have like 100 to 300%, so 1 to 3 times larger deformations in the vertebrae with lytic lesions. For mixed lesions, you have cases where most of the cases, again, the vertebrae with the mixed lesions have bigger deformation than the adhesion control. For the blastic lesion, actually, it's another story. So you have actually almost similar strain or similar deformation, except in one case where we had even higher deformation in the control vertebra compared to the one with the blastic lesion. So just to tell you here that the situation is complex, but we have now tools to better understand, to measure the deformation of this vertebrae. And not only, but we can use this tool also to validate the output of computational models. And that is what I'm going to show you in the next couple of subtopics or points in my lecture. So in the first part, I would like to talk about some in silico approaches that we could use to evaluate the effect of the properties of the lesions on the biomechanical properties of the vertebra to try to improve scoring systems. So first, we did a study to validate high-resolution microfine entanglement models of vertebrae with lesions. So what are these type of models? So these are models that can be based on high-resolution micro-CT images. So these are lab scans. It cannot be done in vivo at the moment. But we can still learn about what is the effect of the lesion properties, as I will show you in a bit. So we can start from this image here. We can then create a fine entanglement model by just segmenting the image, understanding what is bone and what is not bone, and converting every single voxel of the bone into a fine entanglement, which usually are linear hexahedral elements. So these are very computationally expensive models that can be run in a high-performance computer, but they account for all the microstructure of the tissue of this complex bones, in this case, the vertebral body. So we did a bit of a study first where we used the information that we got from the experiments on the porcine vertebrae, where we did exactly this micro-CT scans from the undeformed sample, the micro-CT scan of the deformed sample, and as I showed you, we could identify the displacement maps. And then from the undeformed configuration, we created our finite element models. We matched the boundary conditions as it happened in the experiments, and we compared the results to do validation. And we did that in this case for the porcine vertebrae, both intact and after we induced the lesion. These are, as I said, these are very expensive computational models. So we usually do linear models, both in geometry and in materials, which means that we can't really predict the nonlinear failure behavior of the tissue, but we are more interested in understanding actually what happens where the deformation localizes in the early stages of the load, of the application of the load. So we did compare here digital volume correlation measurements. So these are our gold standards. So this is actually basically is our real measurement. And we compared it with the output of the finite element models that was done only in this initial region to exclude the growth plates that are still open and unmineralized in the bone, in the porcine tissue. And we compared then actually, for the different samples, we compared the displacements from the models with the displacements of the DVC outputs of the experiments. And we could see that actually we got very good predictions for the intact case and good predictions for the lesion case, but with some scatter, in particular for one of the samples. As you can see here, in one of the samples, we had quite a bit of scatter from the one-to-one relationship, which is actually where we would expect this prediction to fall. And the problem is that in this case, we identified that there is a localization of the deformation and after yield, basically, so in the nonlinear part of the load displacement curve in regions which are very close to the lesion. And that is exactly what why probably our linear model are not able to catch this, at least the deformation around those points. However, in general, it worked pretty well. And also for most of the samples, even after the damage, it worked pretty well. So we said, okay, well, now let's use these models to learn more about the properties of the lesions in terms of size and location. So we did scan a number of human vertebrae. These are healthy vertebrae from donors who died of causes which are not associated to musculoskeletal diseases. And what we did is that, but we did micro-CT scans of those, and then we started simulated lytic lesions like holes in the structures of different size, so like 4% of the vertebral body volume, 12%, 24%, and 35% of the volume. And we did simulate them in different regions. So in the lateral right, lateral left, posterior side, anterior side, and central sides. So that from the same model, we can look at the distribution of the deformations and the mechanical properties when in the undeformed control model, or in cases that there is one or more lesions simulated in different locations of the vertebral body. And the question here was mainly, does the location or the size of the lesion play a big role, so that we could potentially ask our clinical partners to include that in the scoring system. So we look at the strain distributions, and we also estimated the local stiffness from this high-resolution, large computational models, and we looked at the difference or the reduction, in this case in failure load, but very similar trend was found for the stiffness, the reduction in the mechanical properties in function of the size of the lesion and the location of the lesion that you see here as central, lateral right, lateral left, posterior, and anterior. And you can see here is that while the volume of the lesion plays almost a linear, has also a linear relationship with the reduction in mechanical properties, the location of the lesion has a minor effect. I mean, in the very large lesions, there is a bit of difference between lateral left and, for example, central. In this case, this is the biggest difference that we could see. But again, this is probably due to, well, it depends on the structure of the vertebra. If we compare then the single lesion with the multiple lesions, but with the same volume, so the sum here, it's still 4%, so it is 2% in this side and 2% in the other lesion. We see that actually the reduction is pretty similar than the single lesions. And again, the location with all the different combination doesn't play a big role. What plays a big role is the volume of the lesion. So to conclude here, we showed that the lesion volume should be included in scoring system is likely to have a big impact on the vertebral strength, while the location and the single versus multiple lesions seems to have less importance. Now, I want to say seems to, because actually in order to generalize the finding, we need to go back to our modeling approach. And instead of simulating only a uniaxial compression, as we did in our case, we would need to actually apply different loading scenarios. And also, as you can see here, we took also only a vertebral body slice. We removed the end plates. Again, we would need to include the end plates also in our assessment. But it seems that the volume of the lesion is the one that is mainly affecting, and it is relatively easy to measure from the CT scan. So it is definitely a score that could be added. Right, so the second part, I would like to talk about more of, actually these are in silico, computational models, biomechanical models that can be based on the biomedical image for a certain subject. So these are subject-specific models. And what we did first is that we did a very similar validation study compared to the one that we did with the porcine vertebrae, but on human vertebrae, and instead of using microfine element models, using what are called homogenized fine element models, which are typical models that we can build from biomedical images. So this was done in collaboration with the group in Bologna, so with Professor Marco Viceconti and Chiara Garavelli, who was doing her PhD and did a visit in Sheffield. And what we did actually in Sheffield is that we performed this mechanical testing, as I showed you, with Marco Palanca, where we did this stepwise loading, and we did the micro CT images of every single step. And then we did from the unloaded vertebrae and the image of the loaded vertebrae, we calculated our digital volume correlation maps. And from the undeformed image, we created what are called homogenized finite element models. So these are finite element models which are much less computationally expensive than microfilm models, and where the material properties are based on the local density distribution that can be acquired from the images. Now, the caveat here is that we still did the analysis based it on high resolution images by, basically having a sort of an equivalent of subsampling of the image resolution in order to create images which are similar to CT images in order to inform these models. But in reality, the question was how far would be these models if we would base them from real clinical CT. So in reality, for these samples, we had also clinical CTs. So for one case, just as a sanity check, we did compare the displacement field that were based on models from clinical CT images and those based on models from micro CT images and of the same model, of the same loading condition. And as you can see, actually, the displacement field is very similar in all the three Cartesian directions. So in reality, we are quite confident that these models based on micro CT behave similarly to the models that we could base from clinical CTs. So again, going back to our approach, what we did is that we have our experimental digital volume correlation map of the displacement in every vertebra. From the undeformed, we can create our homogenized finite element model, basically assuming heterogeneous material properties based on the local bone mineral density. And then we matched the boundary conditions on the top and the bottom end plates to make sure that we are loading them in the same way. And then we can look at the strain field from the DVC, from the experiments, and the strain field from the microFE, and then compare the strains and the displacement. And as you can see here, these are just plotting the FE displacement and the DVC, so the experimental displacement, along the crinocaudal direction, so in the axial direction. And in average, actually, the models are doing a good job. It's a bit worse than what we got from the microFE models, but still, it can predict pretty well the local deformation, or the local displacement, I should say, both for control, lytic, and mixed and vertebrae with mixed and lytic lesions. If you look at the other two loading directions, actually, or the two Cartesian directions, so the anteroposterior direction and the mediolateral direction, actually, we did a bit worse but than the crinocaudal direction, simply because usually the range of the displacement here are pretty small, because we are testing everything within the elastic regime, and we're trying to predict with our linear model the deformation. So again, the displacements are pretty small. The errors are of the DVC, which is our experimental technique, also are a bit higher with lower displacements. So again, we are trusting more the output from the crinocaudal direction. If we look just at the correlations between the displacements for the magnitude and for the three Cartesian directions across all the different specimens, we see that for a few of them, actually, we get very bad correlations. And these are usually due to the fact that the deformations or the displacements are extremely small, or that there were some nonlinearity, some localization of the deformation in those vertebrae that could not be catched well by our models. But just in fairness, I wanted to show you that it is not every time leading to excellent correlations, but in most of the cases it does. And we can definitely trust this type of models to look at the local deformation in these complex heterogeneous structures. So by looking at the distribution of the minimum principal strains, so these are, again, the local principal deformations. These I just report three cases. So the DVC on the top, these are the experimental measurements. The FE on the bottom, these are the homogenized FE from the biomechanical models. And from the left, there is a control vertebra. In the middle, a lytic vertebra with a lytic lesion. And on the right, a vertebra with a mixed lesion. And as you can see, in terms of deformation patterns, we can get pretty well the control vertebra and the deformations in the control vertebra and those with lytic lesions, but actually we don't do as good job in the vertebrae with mixed lesions, which is probably where we needed to improve how we assign the material properties, but this is a work in progress. So once we were quite confident that our models can predict pretty well the deformation and the mechanical properties, actually we went also on and did a feasibility study on real patients. So these are in collaboration with the National Center for Spinal Disorders in Budapest. They shared 8 CT scans from patients who had vertebrae with lytic lesions, and with a SINS score, so this clinical score in the indetermined regime, so basically range. So basically where, again, the decision was going completely back to the experience of the surgeons and of the oncologist. So we had like 3 males and 5 females with this age here. And what we decided to do is that for every subject, we modeled the vertebra with the lesions and the 2 adjacent control. So the question here was, is the vertebra with the lesions more or less critically deformed compared, or is it more critical than the adjacent control? If it is more critical, it's likely that there is that this actually something needs to be done. If it is as critical, as strong, or even stronger than the adjacent control, then our pipeline would tell that the clinician that it shouldn't actually be critical. So just a few words about the modeling pipeline to expand what I just was saying before. So we start from a QCT scan. We do a manual segmentation of the vertebra in this case to identify the contour of the vertebra. We do an alignment of the STL file. We do a smooth mesh. So here we use a smooth tetrahedral nonlinear, actually quadratic element, so with 10 nodes. And then we apply a compressive load on the vertebral body, and we define, according to literature, the failure as the load that is required to deform the vertebral body of 1.9% of apparent deformation. So, of course, this requires a calibration of the CT scan to make sure that we know what is the density. This is done through a calibration phantom, and this allows us to evaluate the local density everywhere in the point and in every element, and based on that, we can assign the material properties based on this set of equations that convert the CT value of the gray levels, basically, into a local elastic modulus and a local yield stress and yield strain in function of the apparent density. And again, if you want more details about this, let me know and I can expand. In terms of mesh, we used the T10, as I said, so tetrahedral elements with 10 nodes with a maximum side length of 1 millimeters, which came from a mesh refinement study that we did on a subgroup of the vertebrae. And these are the type of things that we can look at. Well, first of all, we can look at the densitometric properties because we have calibrated images. So this is the frequency plot on the vertebrae with lytic lesions in T5 and the two adjacent control T4 and T6. And actually here you can see how the lower is the density of the lytic lesion of the vertebrae with the lytic lesions compared to the two adjacent control, which are almost overlapped. So this is already confirming somehow what is the change in bone mineral density. And then from the mechanical point of view, we can look at the stress strain curves. So these basically are the normalized forces and versus the normalized apparent displacements. And we can look at the stiffness or the Young's modulus, and we can look at the failure load, the yield load, or the failure stress and the yield stress, and the energy to failure and the work to failure. All the details, if you want, are in this paper here in JMBBM in 2019. But just to give you an idea, again, this is a busy table. I don't want you to read the values here. It's just to tell you that if we look at the bone mineral content and the bone mineral density, so how much bone is in each vertebra, and if you look at the mechanical properties, the structural one or the normalized ones, and we split between the control vertebrae and the ones and those with lytic lesions, we see that basically there is no difference. So it has basically no sense to look at this patient as a group. We need to really look at case by case what happens and how critical is the lesion case by case. Similarly, if we plot here all the load displacement curve that we get from our models for the vertebrae with lytic lesions in red and the control vertebrae in black for every patient, again, we can't see a clear threshold where we would say, look, below this certain stress is likely that the vertebra will fail, but we need, again, to go case by case. We also look at the correlation between the strength, so that is the ultimate stress, and the SIN score. This is the scoring system for that vertebra. And we see that there is a trend, but this is not a significant correlation, which means that actually our models are telling more than the clinical tool. Of course, I would be interested in looking at this again whenever we have big numbers, because we had only 8 cases and in total around 10, 12 vertebrae with lesions. So as I said, we need to go back and look at case by case. So this is what we did. So for example, for this case here, we had a patient with a renal carcinoma, and they had 2 lesions. So one in L1, as you can see here, and one in L4, which is here. And basically and it was very hard to understand what was if these vertebrae were critical or not. So we modeled also T12, L2, L3, and L5. And these are the results from our mechanical testing or the computational models. We can see here in red the stress strain curves for the lytic lesions, L1 and L4, and in blue and black, the control vertebrae, T12, L2, L3, and L5. And we can see that actually the vertebrae are less strong and less stiff. So the vertebrae with the lesions are less strong and less stiff compared to the controls, and in particular around 20% of difference in all the different mechanical properties. So for us, I would say that from our tool, will come up that this would be a FE or biomechanical warning. So we would tell back the the clinicians that biomechanically the vertebrae with the lesions were less stable than the adhesion control, and therefore probably something needs to be done. In another case here, there was a patient with a chondrosarcoma with the lesions in L4. And again, you see also a sort of densification around the lesion. And in fact, actually, when we compare the two controls, L3 and L5, with the vertebra with the metastatic lesion in L4, we saw that the metastatic lesion even was stronger and stiffer than the adhesion control, which were almost overlapped here. So again, in this case for us, this would be a less biomechanically critical compared to the other case. And finally, there's another patient here that actually different types of lesions in T12 and L1, in particular one what was very focal and localized, the other one a bit more spread. And in this case, for example, we could see that the vertebra, so the vertebrae with the lytic lesions, one was more critical than the controls, and the other one was less critical. And in particular here, we could see that we had an FE warning, biomechanical wording for the vertebra T12, which is the one with a very, very focal lesion. But due to the location and the gradient of density was even more critical than the vertebra with the more spread by lesion in this case. So again, this is another tool that even in the same patient, you could see, you could use this tool to look at which is the most critical vertebra. So I think that I don't have the time to go through this, so I will just close the talk with a few perspective and a few words about the Metastases project, which is a project that we are currently running. So in terms of future perspective, I hope that I told you that, well, I made the point that we need to learn more about the biomechanical properties of this very complex vertebrae with lesions. I really believe that combining data-driven, so AI tools and subject-specific models would be beneficial in this area, but we need very large numbers, and I will come back to this in a minute. And we need even more validation studies for very complex scenarios, in particular for loading scenarios which are paraphysiological or which are combination of compression, bending, and torsion. To do so, we would need probably to model also the intervertebral disc. So everything that I told you today is about modeling the vertebra, but the load is transmitted through the intervertebral disc. And we have seen how the failure modes are related to a failure close to the end plate, which means that probably it is also due to how the load is transferred through the intervertebral disc. So it is very important to study that. And finally, it would be great to have some predicted models of bone adaptation over time, but we make the things even more complex, and I will come back to this in my last slide. So in order to tackle most of these points, so the first 4 points, actually we have started a project. It is funded by the European Union. It's called Metastra. It is to transform the fracture risk prediction for cancer patients with vertebral metastases. It is an Horizon Europe call with contribution from UKRI and the Swiss National Science Foundation. It is coordinated by Bologna, by Luca Cristofolini, and it started in 2023 and with around 8 million euros of budget. And the idea is actually to put together, so we put together a big team of people from different centers here in Europe. In particular, I want to stress that we have 4 clinical centers, including Rizzoli in Italy, Eindhoven in the Netherlands, the National Center for Spine Disorder in Hungary, and the Charité Berlin in Germany. And these are collecting a lot of clinical data that are then are needed to inform and validate computational models, both data-driven and physiology-driven models, which are built in Sheffield, Zaragoza, Zurich, Szeged, and with the help of in silico trials. And of course, Bologna will have a key role also in not only in coordinating the project, but also in delivering advanced mechanical testing for the biomechanical validation of these models. So if you are interested, this is on the bottom left, you can see the website where there's much more information than the one that I can give you in a couple of minutes. But basically, we have actually our package, which is basically aims at collect a lot of data retrospectively on a number of 2,000 cases that can be used to train our data-driven models. And then we will develop our physiology-based driven models in Sheffield with the help of the other partners in the consortium to evaluate the biomechanical properties of spine segments. And we will do an extensive validation, verification, and uncertainty quantification against experiments in Bologna. And finally, we will do a prospective study with 200 clinical data sets. And this will provide us with a decision supporting system that is based on basically assessing first the fracture risk in patients with the clinical tool, so the SIN score that I mentioned before, and then using explainable AI to predict the risk of fracture in patients with the data-driven models, and then in the more complex cases, build biomechanical models to evaluate the risk of fracture and also to evaluate the effect of treatments. I think that I stop here because my voice is gone. But just thanking my team and the Integrated Musculoskeletal Biomechanics group, in particular Maria Cristiana Costa, Marco Palanca, Giulia Cavazzoni, and also Chiara Garavelli for the work done and getting the data that I showed you, and all the funders and you for your attention.
Well, many thanks for this talk and giving this real overview of different type of research, different starting from also what is interesting, was interesting for me is this experimental, let's say, work here. So it is really, it was really challenging. Well, we have time for questions and suggestions. I would like to start with some questions about some details about finite element method you are using here. So how many of these elements you really need, and probably it is a challenge because of the structure of bones. So it's if you could give some more information about this.
Yeah, of course. So as you're saying, the problem is that the structure of bone of the vertebra already on its own is very complex. On top, we have the problem of the lesion, which is making it even more heterogeneous. So we have used the two types of modeling approaches. So one modeling approach that I mentioned based on high resolution images, these are starting from a very large to a very detailed high resolution image that tells us all the microstructure within the tissue. And we can really look into it and model it in details. When I said that basically we are able to segment the bone structure and then model every single bone voxels into a finite element, means that we are usually dealing with more than 100 million elements. So we are talking about hundreds of millions of elements, which makes the model very, very computationally expensive. On the bright side is that usually you have such a detailed microstructure that using linear models is usually enough, because at that dimensional scale, bone behaves relatively linearly until it reaches a very high deformation and then it fails. So usually you can use linear models in this case, and therefore by using an HPC that we have, for example, in Sheffield, but which is not huge, we can still run it in around half an hour or so. But we're talking about a very large number of elements. The other ones, the clinical models, so those that are based on CT scans, so that I showed you in the last part of my talk, those are much less expensive because we can build tetrahedral elements with the 10 nodes. Apologies. And in this case, basically what we can do is we have around a few 100,000 of elements. But then the tricky part is that in that case, as we are not modeling the whole microstructure, but only the density, these differences in density in different regions of the bone in an with an homogenized approach, we need then usually to take into account for local non-linearities, material non-linearities. So even though actually it is a much lower number of elements, we still run it usually in HPC just because of the non-linearities that we have, and therefore the simulation takes a bit longer.
Well, so you have already touched the problem of non-linearities, because yes, probably in the case of deformation and changing, let's say, material properties, it is probably a challenge, yes, to take.
It is, and so there is a lot of work done. I haven't had the time to go through it, but there's a lot of work where we can link pretty well how a healthy or osteoporotic, so we'll say benign tissue, benign pathologic tissue, can what is it, the link between the bone mineral density and the elastic modulus, so the linear component of the mechanical response, and also the yield properties and the failure properties. So we have a lot of experiments done in the past by several groups worldwide where we know pretty well what are those relationships. The problem, the big question is what happens when the lesions come in. When you have even more heterogeneity, especially in the mixed lesions, where you have a lot of heterogeneities and gradients in mechanical properties within the structure, and if you need to take that into account with a complex nonlinear model, and if we needed to adjust those phenomenological law that links the bone mineral density with the mechanical properties at the local tissue level. For us, at the moment, the approach is that we are not changing it. So the idea is that the lesion will affect the density distribution, and by catching well that, we will catch well the material properties. It is a big assumption, but the good thing is that now we have experiments to challenge this assumption. So we can really build the models by standard constitutive laws or a bit more complex constitutive laws and see if we are able to catch the bone mechanical properties that we measure in the experiments, which is very important.
And I understand that it was mostly about bones, but how you can take into, let's say, into this consideration also this muscular part, it is by boundary conditions or?
Yeah, so usually, well, the spine is very complex, and in reality, you have bone that is the one that fracture. In this case, that we are really interested in. That's why we model it very, very, very in details. But you're right. So the same that basically what really will play a role are the boundary conditions and how we can model that the effect of the deformation in the other tissues and the muscle forces and the deformation of the intervertebral disc and how it distributes the forces into the vertebral body through the boundary conditions. So at the moment, we're using a relatively simplified tool, ways of assigning the boundary conditions in compressive loads. But in the Metastra project, we are developing actually together with the other colleagues across Europe, the model of a spine segment. So where we have a vertebrae and also the intervertebral discs, and we have the muscle forces that are attached to it and that will drive the deformation of the tissue. That is much more complex, but that is one we are going to tackle in the next 4 years.
Maybe it will look like a stupid remark, but well, I think that in all of this car industry, they have developed a lot of approaches to how to use finite element methods for deformation. So, do you have some, let's say, do you think that there will be some inspiration for your work? Or?
Yeah, I mean, honestly, we are using usually static models, so we are using implicit methods, and in most of these cases, it's fine to look at relatively slow phenomena. And the vertebral fractures are relatively slow phenomena because you have a local deformation which accumulates. And of course, getting approaches from engineering has been the initial inspiration, right? So we replaced the CAD design with CT scans. We have done a similar mesh and then applied the material properties, which is the very complex part in biological tissues. So the inspiration is already there. What I believe there will be even more possibilities in the future is to look at more dynamic processes. It's something that we haven't look into it in details right now. And in some cases of some type of fracture, like vertebral fractures due to falls or trauma, there are colleagues, especially ETH Zurich, who have done or started using explicit methods where you can really look at the dynamic process. And that is, of course, the car and aerospace industries can be of inspiration for that. The problem that we have are that these are not materials that we design. The
yes.
materials are, we need to learn about the materials, and the diseases, of course, affect those. So we need to go back in the lab and measure the properties before we can really model them properly. That's the big challenge for us.
Let's hope that the material was designed well. Depends on persons by evolution or somebody who knows.
Usually, yes, but then whenever you have a disease, then it's another problem. That is where you go outside the population, right? So the typical data that you would expect in a population, and that is where, in particular, we have seen that this metastatic disease, it's so different by patient, case by case, that is very, very difficult, if not impossible, to generalize. So you need really to look at, especially in the difficult cases, you need to look at subject-specific personalized models to tackle the problem. That's what I believe.
Yes, and also the structure of this material is also not so trivial. So it is taking all this into account is very difficult. And well, the question is about the link to clinical practice. So I understand that what you can do now, you can help to decide about how it looks from the point of view of a scoring system. Yes.
Both. So I think that already we mentioned we informed the clinical practice, but again, this needs still to be changed. That adding information about the size of the lesion and to some extent the location of the lesion, but as a second priority, would be important to be taken into account in the scoring systems. And I showed you some other studies have been done also in Bologna and other areas where they show the importance of that. So still, we're going to try to push the clinical community to say, look, you need to take that into account. But the other thing is more doing this sort of subject-specific model. So, sorry, I lost the voice exactly when I had to talk about this, but now that it's back, I can expand a bit. So in the Metasta project, what we try to do is to create a decision support system, which is based on different levels. So the first level will be the SIN score. So this is basically the standard scoring system in clinical one, but we'll try to automatize that. So automatically assess that from the biomedical images so that the clinician doesn't have to identify the scores himself. The second level is that we are going to use a well-trained Metastra AI model that we call, so basically it's a data-driven model that will be fed and trained with 2,000 cases of fracture and not fractured cases that are collected exactly at the moment by our clinical partners. And hopefully, so if you remember this, one step back, if you remember this SIN scores as a big gray zone. So a zone where actually the clinical tools say, well, I have no clue. You need to go back to the experience of the surgeon. So we hope that with the Metastra AI, this gray zone will be reduced because we are getting more information from the biomedical images, from the patient's information, characteristics, age, gender, country, level of activity, and so on. But still, we believe that there will be some very difficult cases in the very middle that will still be gray. So where the Metastra AI will not provide a proper advice. That is where we can do the subject-specific biomechanical models for those cases. These are more expensive. These are more, but are more detailed. And that is where, hopefully, with these 3 tiers decision support system, we could go into the practice. Of course, it needs validation, it needs certification, it needs to go through all the processes of converting a modeling pipeline into a certified medical device. And in Metastra, we will not sort all of this, but we will hopefully do the biomechanical validation, the clinical validation with 200 prospective studies, patients, and then we will have a roadmap towards certification. Which hopefully will then jump into the clinics in the next 10 years,
Yes,
something like that.
it's really challenging in the sense that for many people it is a decision about what to do in the case they have such a disease is really crucial. So sometimes it is just something what can be removed by, let's say, a kind of physiotherapy. And it probably it is very, sometimes it's all these symptoms are very similar to the case when you need really a kind of operation. So it is something very, very important.
And not only, I mean, I think there are 2 points here that I'd like to discuss. I mean, one is these biomechanical models then could simulate different types of interventions. So the 2 types of surgeries that usually are done in the case that the spine is found to be unstable is either an open spine surgery with rod and screws. So basically, you really fix or make a bridge between the 2 healthy vertebrae to avoid loading into the metastatic vertebra. Or there is another component where you can inject cement, like a sort of vertebroplasty. It's called inject cement to fill the holes. And again, in many cases, this is going back to the decision of the surgeon, depending on of the spine surgeons, depending on their experience. But our models can tell for that particular case, actually, which would be the best case scenario, which would be the best treatment. And the second point that I wanted to make is that I mentioned several times that in the standard practice, we are going to, the decision is going to be back on the shoulder of the clinicians. And this depends extremely on the experience of the clinician, of the center where they're working, on the experience and training that they went through. So in order to, I see this decision supporting system based on computational models as a way of democratizing, if you want, the way that the patients are managed. And that's, in fact, actually why we, in Metastra project, we picked 4 clinical centers, which are actually the best that you can get in Europe, probably in this area, in the area of spine metastasis. And if our models is able at least to replicate what would be their decision, it would be still a great success, because it means that then this could be used in smaller centers across Europe and the world to actually help the clinicians to take the right decision.
Yes. Well, we have already reached the time devoted to our seminar, but well, I know that you are coming to Krakow for SanoScience Day, so I think that there will be also an opportunity for discussion with our colleagues mostly from scientific programmers team who took place in Silicon World, led by Marco. And of course, this bone strength simulation was one of, let's say, key application of the system we have developed here in Sano. So, Enrico, many thanks for your talk. Many thanks for taking part in discussion. And well, we are looking to see you in, let's say, in reality in Krakow. Thank you very much.
Looking forward for the next week. Thanks a lot for the invitation and for the discussion, and see you soon in Krakow next week.
Thank you very much. And thank you very much for everybody who decided to join our seminar. The next, let's say, seminar, to some extent, probably will organize the transmission of SanoScience Day. It is not clear how it will be done. We will let you know. But anyhow, the next 3 seminars will be devoted to Nobel Prizes in chemistry, in physics, and in medicine. So, so many thanks, and see you on Sano Seminar in 2 weeks from now. Bye.